KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, now in this video we are discussing continuation information of centrioles and centrosome, okay, or centrosome and centrioles. Apart from that, later we will discuss about the microbodies. Later new chapter I will start. Okay, now let us have a look on the final information of cell cycle and cell division chapter. So there last and final information regarding centrosome and centrioles. Okay, now centrosome is an cell organelle or organelle usually containing two cylindrical structures. Centrosome is an organelle usually containing two cylindrical structures called centrioles. So means if you take a centrosome, there two different types of two cylindrical structures are present within the centrosome. So these two cylindrical structures we will be considering as, or we will be calling as, centrioles. So this 2 or 3 lines information so I have given in a previous video also. So centrosome is an organelle usually containing two cylindrical structures, so called centrioles. So they are surrounded by amorphous pericentriolar materials. This is NEET examination point of view important. So definitely what are the centrioles, or one pair of centrioles means two cylindrical structures are there, no? So definitely all are surrounded by amorphous pericentriolar materials. Amorphous pericentriolar materials. Both the centrioles, so here we have taken two cylindrical structures, centrioles are there. So these are two centrioles in a centrosome lie perpendicular to each other. So definitely all these are all like arranged like perpendicular to each other. Okay, perpendicular to each other, in which each has an organization like a cartwheel. So there and each and every one, so means here two are there, no, there in which each has an organization like cartwheel. So means if you take one particular structure, so you are observing one diagram here. So this particular diagram if you see there, no, so you could not find even in our textbook also. So it is apart from the textbook. So there each and every centriole structure it is being like a cartwheel. Now they are made up of 9 evenly spaced peripheral fibrils of tubulin proteins. One more time you see there. So how it is, this particular cartwheel, how it is made up of means mainly made up of 9 evenly spaced peripheral fibrils. 9 evenly spaced peripheral fibrils are there. Once you see once you look at the diagram exactly, let's spotlight, let us count from there. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so 9 evenly spaced peripheral fibrils of tubulin protein means here these peripheral fibrils are made up of tubulin protein. This is important, tubulin protein. Next one, each of the peripheral fibril means if you take one peripheral fibril, definitely that ends with a triplet. So that's why here we have written like this, each of the peripheral fibril is a triplet. So in the end of the once you see that, this one peripheral fibril, likewise 9 are there, no? So end of this peripheral fibrils, so 3 triplets, 3 means tree only, no? TRI, tree means like a triplets, means 3 are there instead of 3, no? So we can together call it as a triplet. Bi means 2, mono means single, tri means 3, no? So likewise, so 3 microtubules together present, no? So that's why this particular structure we will be considering as triplet. Ipo these triplets where we are observing in the end of the peripheral fibrils. Purida. Next one, the adjacent triplets are also linked. So means adjacent triplets. Ipo adjacent means pakkadalidana. So ipo paringa inge idu triplet. Once you see there ellame triplets ellame link apadana linked means joined aichi, so connected. So this one. So what is a bridge like structure, or what is a connection is there, no? That particular connection name is known as AC linker. One more time, once you see the diagram, இப்போ கொஞ்சம் க்ளோஸாக பார்த்துக்கோங்கம்மா. இப்போ பாருங்கள் ABC, இங்கே 1 microtubule, that's why 1 microtubule, second one, third one, that's why here we have given names as ABC. இப்போ பாருங்கள், இப்போ A and C are connected together with the link, and C and A, that's why A and C, that's why this particular name, you know, this exactly, let's spotlight one more time. You see this particular place or line, you know, we are calling as AC linker means, so that particular link, you know, which joins the A triplet and another triplets C1. Okay, so thereby that particular thing, adjacent triplets also linked. Next one, the central part of the proximal region of the centriole, or simply you can remember like this, that is a central part of the centriole is also proteinaceous, means that is also made up of with proteins. Okay, that particular name is known as hub. Okay, so central part of the proximal region of the centriole is also proteinaceous, means here central hub. Amma idi central hub amma. Idi exactly, let's spotlight we are seeing now. So idi hub, hub. Okay, that particular hub also is made up of proteins. Next one, which is connected by tubules. Puridama, so which is connected by tubules of peripheral triplets. So ipo idellame connect aichidana. So triplets all are connected with the hub, isn't it? So by radial spokes, so here even radial spokes also we are observing. So by the radial spokes, so thereby here what is the point here we can understand, no? Totally how many number of radial spokes are there? 9 radial spokes are present. Okay, so their peripheral triplets, one more time you see there, which are connected with tubules of the peripheral triplets by radial spokes, made up of, even radial spokes also are made up of proteins. Okay, next one, centrioles form a basal body of the cilia. So now here main important explanation we are entering now. So suppose centrioles are there, no? So where these kind of centrioles help us means, these centrioles form basal body of cilia or basal body of flagella. So here while explaining cell the unit of life chapter introduction, so we have seen flagella structure. If you see the flagella structure there, 3 parts are there: hook, basal body, emma. So definitely their particular basal body is made up of centrioles. Okay, so thereby basal body of a flagella or the basal body of a cilia, so definitely there is involvement of centrioles for the formation of. Okay, now next one is spindle fibers, so that gives rise to spindle apparatus. Not only these centrioles form the basal body, so that is one kind of a function. Next one is spindle fibers, that gives rise to spindle apparatus during cell division in animal cells. Means here these centrioles involves in the development of spindle fibers Where? So definitely in the cell cycle and cell division, only in case of animals. So thereby centrioles definitely involves in two important functions. One is basal body formation, later spindle fibers that gives rise to spindle apparatus formation. So this particular spindle apparatus formation we can observe during cell division in animal cells. Okay, so likewise somewhat information, so we can discuss related to the centrosome and centrioles. Ipo parunga, here three centrioles are there, the three microtubules are there, together we are calling as triplets, and the central one is a hub. Next one, this one is a radial spoke. So 1 2 3 4 5 6 7 8 9. Okay, radial spokes, so 9 radial spokes are there. So thereby once now let us discuss, let us compare, not discuss, compare. So which one structure of cilia or flagella. So in a previous class we seen, in a previous video, there number of radial spokes 9 we seen. So even in a centrosome also, or in a centrioles also, centriole, in a centriole also we are observing number of radial spokes 9. So next one, triplets here, how many are there? 1 2 3 4 5 6 7 8 9 are there. So 9 into 3. So in each triplet, no, 3 microtubules are there, no. Suppose in a NEET examination, if they ask a number regarding triplets, means 9 into, means if they ask regarding microtubules, so there 9 into 3 we have to put. Why? Because triplets, so 3 over there, microtubules, so 3 together we are calling as triplets. So that's why number of microtubules here 9 into 3, totally 27 amount. So in case of structure of a cilia or flagella, so there doublets only over there, no. So thereby there 9 into 2, so 18. If ratio of these two, so there 18 and 27 means 2:3. So that particular thing maximum understanding pannikonga, otherwise I will explain when you will be here. Okay, no problem. So that is the information regarding centrosome and centrioles. In this particular occasion, so what is the information we have discussed means, so suppose if you take one particular centrosome, there centrioles are there. So these centrioles also, how many are there exactly at spotlight? So we can get the answer. Two cylindrical structures are present. These structures only we are calling as centrioles, and these are all covered by means of amorphous pericentriolar materials. So there these centrioles also arranged perpendicular to each other. Mathematics point of view, 90 degrees angle, somewhat we will be calling, no, so that one. And if you take each and every centriole, no, that particular thing being like a cartwheel. So next one, now till now we have seen the information of like centrioles. Okay, so this is a structure, 1 or 2 times try to practice it. Next one, now last and final conclusion, that is microbodies. Microbodies point of view, many membrane bound minute vesicles. One more time, many membrane bound minute vesicles called microbodies that contain various, so that contain various enzymes, present in both plants and animals. So microbodies, the word itself giving information, no, microbodies means seems to be very small, isn't it? So many membrane bound minute vesicles, very minute vesicles are present in a cell. So such kind of minute structures, so which contains various types of enzymes. So definitely these kind of structures we can present, or we can see in both plants as well as animal cells. So that what are the kinds of microbodies, sir? Try to give any examples means. So example, for example, peroxisomes and glyoxisomes. So still so many we can be taken. Okay, so peroxisomes and glyoxisomes. So why I have taken these two means? So these two are mainly. So we will discuss in the next coming chapters. So that's why these two I have given. So one is peroxisomes and one more is glyoxisomes. So this is a small information regarding chapter name is cell, the unit of life. Okay, now new chapter I am going to start. New chapter number is 10 and the chapter name is cell cycle and cell division. Okay, cell cycle and cell division. So that is the information now. Now what is information or what is introduction class here we can discuss means growth and reproduction. Growth and reproduction are the characteristics of cells. So means definitely if you take particular cells, no, growth and reproduction are the characteristics of cells. Here we no need to discuss anything, so we can understand it clearly. So indeed of all living organisms. So correct only, no. Next one, all cells reproduce by dividing into two. So definitely when the cells are going to be divided, so all cells reproduce by dividing into two, which each parental cell gives rise to two daughter cells each time they divide. So thereby what is the point? When the suppose if the cells are going to be divided at the time, no, each and every parental cell that can gives rise to two young ones. That can give rise to two young ones. So thereby here what is the point we are discussing now? Suppose if one parental cell is going or undergoing for division means, so that particular parental cell definitely gives rise to two daughter cells whenever it will divide. Okay, so thereby what is the point here we are discussing means, suppose if one parental cell is going to be divided, definitely by that division, definitely two daughter cells of formation takes place, or definitely two offsprings of formation takes place when they divide. Next one, these newly formed daughter cells can themselves grow and divide, gives rise to a new cell population that is formed by the growth and division of a single parental cell. So means here what is information again we can or we have to understand means, so what are the daughter cells are formed from the parent. So again these daughter cells so grow themselves, so definitely grow themselves and divide. First of all, so one more time I'll explain. Once you see one parental cell irukama, so that particular parental cell divide. Okay, so by the process of cell cycle and cell division it divides. So at the time two daughter cells of formation takes place. Now these two daughter cells grow. So first of all, so growth process everything, later divide. So divide and gives rise to new cell population. So thereby one cell 2 cells of 4 cells, 4 8 16 likewise one complete cell population formation. So it means here definitely how sir this one cell how sir it will be divided like this means, so by the process of growth and cell division of a single parent. So thereby single parent and its progeny. So thereby first of all parental cell are going to be divided, later progeny going to be divided, and finally so by the growth of cell and division of the cells, one complete cell population formation takes place. The group of cells only later we will be taking as tissue. So thereby here in this particular case tissue formation and everything so definitely takes place or formation takes place only because of the growth and division of the cells. That particular cells may be parental cells as well as progeny cells. Next one, in another words, so which cycles of growth and division allow a single cycle to form a structures consisting of million number of cells. So thereby in another words what is the information we can discuss. Such cycles of growth and dividing. So means here when the cells are going to be when the cells are going with the growth and when the cells are going to be divided, so thereby what is happening, so million number of cells of formation takes place from the single cell. Simple information, what is the content or the concept means, so what are the cells are going to be divided. Now all these cells are definitely forms group of cells. Group of cells. So how sir means, so first of all that particular cell divided, so means that will grow. Later once it attains maturity, that particular cell will be divided. Okay, so that is the information only. So next one, cell cycle. Next one is cell cycle. Now cell division is a very important process in all living organisms. Important point. So suppose if you take a cell division, it is a very important process in all living organisms. During the division of a cell, DNA replication and cell growth also takes place. Underline it and so remember pannikonga. So thereby during cell division, during the cell division time, now what is happening means DNA replication process takes place, and we have learned already cell growth process also takes place. So thereby cell growth and cell division, okay, cell growth and DNA replication process both are occurs in during cell division. Means here all these processes means what are those? Cell cycle and DNA replication. Next one cell growth. All together hence have to take place in a coordinated way to ensure correct division and transfer and formation of progeny cells. So thereby here these are all one cycle, or these are all these kind of processes are like one coordination way. So all these kind of processes taking place in one coordinated way. So thereby one particular cell that will be that will grow in its size. Later DNA replication process takes place. Later cell division process takes place. Means here all these processes, when all these processes are taken place in a coordinated way, so we can ensure, that the correct division and the formation of progeny cells. So thereby if you take a cell cycle, so there during cell cycle of a cell, DNA replication process and cell growth also takes place. So there what are the processes here we can be taken means cell cycle, DNA replication and cell growth processes definitely taken place. So after completion of these kind of processes only, definitely one particular parental cell will be divided into two offsprings. Okay, so here in this introduction class of cell cycle and cell division, so what are the main important points we have been discussed means, first of all, characteristics of a cell's point of view. One particular cell if you consider, so it has two important characteristics. So first one is growth and reproduction. Later all cells reproduce by dividing themselves. So how they will be divided means, definitely they will be divided so by the cell cycle and cell division. So there again a division also, two types of divisions are there, So first one is mitotic division and one more is meiotic division. Okay, so now here newly formed daughter cells. What are the newly formed daughter cells are there? These cells definitely divide themselves. So before dividing themselves, first of all these cells grow in its size. So then only they can be divided. So means here the division also, two types of divisions are there. So our knowledge point of view we can be divided into two types. One is like mitotic division and one more is meiotic division. So in a mitotic division, so definitely from one parental cell, two offsprings of formation takes place, and what is the chromosomal number is present in the parents, same chromosomal number is repeated or is present in the offsprings. Whereas in case of meiotic division, if you consider, so meiotic division, so there definitely chromosomal number is reduced to half. Chromosomal number is reduced to half. So likewise, so means here particular cell if you consider that particular cell if you are considering as a parental cell, that particular cell is a parental one. First of all that particular cell it will grow in its size, later only it can be divided. So likewise somewhat information so we can be discussed in introduction class. In remaining explanation onwards important points, and along with the important points somewhat extra points also I will try to give you. Thank you and be safe at home.